for taking time to present to the SBGrid consortium, and welcome to our forum.
Thanks, Peter, and also thanks to Michelle and Jason for organizing the series of events, which I think is very nice, at this time to bring people together and update the community and what's ongoing around the world. And so I'm going to share a story that was really spearheaded mostly in my lab by two students and a postdoc, Carl Muir and Jan Lee. But we collaborated with additional groups at EMVL, Christian Herring's Benjamin Roland's group in the Netherlands, and Elze de Wit. So I'm going to talk about recent structural work that leads into sort of insights how chromatin folding is catalyzed by these two key players, CTCF and cohesin. So the model of how the genome is folded is shown on this slide, where nucleosomes, they bind to DNA polymer, and you get this bead on the string model that then further wraps up into this 30 nanometer fiber. And that has been sort of the prevailing model in the field of how chromosomes get folded into these mitotic chromosomes ultimately. And there are structures on sort of intermediates of that assembly pathway, but it turns out that nowadays we do not think that there's structure beyond the 11 nanometer fiber. So the larger fibers are apparently, the evidence for that is not clear, that exists in cells. And actually, if one goes back to the 1970s, one finds in this paper by Paulson and Lemley that when you take all nucleosomes away, all chromatin, you dechromatize the mitotic chromosomes, the shape is preserved, and despite the fact that all these histones are gone. And so it turns out that the shape of chromosomes is determined not at the smaller scale where nucleosomes operate, but at a larger scale where proteins such as cohesin and CTCF interact with the DNA template and catalyze a folding reaction that shapes chromatin. And in these projects on cohesin and CTCF, what I think we encountered, and the reason why I showed this slide is because I think many in the community will face similar problems, namely that if you're working on these larger complexes, one finds that some interactions in these complexes, there are high affinity interactions through sort of large interfaces that lead to a large buried surface area, polymerases or ribosomes, etc. These molecular machines, which we can analyze by cryoEM. Now, there are additional interactions such as this disordered global interactions that also are high affinity, but it turns out the vast majority of protein interactions in complexes actually turns out to happen at this sort of lower, low micromolar affinity range, because these interactions are regulated and they're quite dynamic, and getting at those interactions with cryoEM is going to be quite challenging. And so what one needs to do is, I believe, have a sort of a hybrid approach where one combines these methods together and analyzes these interactions using different kind of approaches. Now, here's a figure that is generated by a student in the lab where we look at the molecular weight of complexes and the concentration, and at one point the concentration on the cryoEM grid becomes prohibitive because the interparticle distance becomes too small. And so there's a green window of opportunity where one typically operates. But the problem, as I said, for complexes that are weak affinity, where one has to work at relatively high concentrations, it becomes difficult to analyze, to reach the sort of green zone where cryo-EM becomes possible. Because when one looks at interaction affinities, for example, in this motif database, we find that a large fraction of interactions are probably too low affinity to be visualizable by cryo-EM, because they would come apart at the concentrations required for the single particle approach. So many projects we work on, they always navigate that, that space, and so we use a different combination of approaches to study these complexes. Now, one of the main developments to study chromosome folding initially was done by EM, as I showed you earlier, of mitotic chromosomes, but nowadays much of that is done by cross-linking and then deep sequencing, generating libraries and deep sequencing, and which allows you to analyze which fragments have been closed in space, and so that allows you to generate interaction maps of chromosomes, or as shown here. So this is what that has shown is that there are two major mechanisms of how how genomes are organized. One is at the sort of very large scale, and these are called compartments. So one has this diagonal, and then the further out you go from the diagonal, you have interactions. And so the dark color is the interaction frequency, and so there are regions that are high frequency and then lower frequencies. And if we zoom in, then so at the top we are at 10 megabase scale. At the bottom we have 1 megabase scale. At this 1 megabase scale, we see these what are called topologically associating domains. Okay, these quadrants in these interaction maps. And the interpretation is that these compartments arise by biochemical reactions, euchromatin and heterochromatin, that self-associates, so that one has heterochromatin that is more towards the lamina, and euchromatin typically towards the interior of the cell nucleus, and it's being acetylated. One key player is this protein, frequently the acetylates, called CDP300, in which we have an interest. And one idea is that this compartmentalization arises because this acetylation is these factors are limiting, and when transcription factors bring them to target sites, you generate these acetylated domains that then coalesce into these so-called A compartments. But as I said, at the smaller scale, one has these topologically associated domains that are catalyzed by a factor, by this cohesin-CTCF proteins. And they are seen on the schematic here as these gray boxes that are close to the diagonal of these interaction matrices. Another view would be this one. So one has a tab where CTCF sites are delimiting these boundaries of these high interacting domains. And the CTCF sites have the unique property that you only get such an interaction in chromatin when the sites are oriented in a convergent way. So when the red and the green arrow are facing each other, then you form these topologically associating domains. Another feature is that frequently the activity, when one looks at where genes are expressed, correlates with this compartmentalization of the genome. And so that has led to a lot of excitement in the field, because there is sort of this extra layer of the 3D genome, which seems to be important for how the genome is organized, how genes are regulated, how one generates active compartments, etc. And so when one looks at these compartments, sometimes they span active and inactive compartments, but sometimes they are encompass only active or only inactive. And so the idea is by folding up the genome, one would generate sort of insulated neighborhoods where enhancers and promoters contact each other, and so explain a lot of long-standing biology about gene regulation and how that the gene regulatory state or the transcriptional state and compartmentalization of the genome are really coupled in an interesting way. Now, if we look into this in more detail, then in the schematic, what we think is happening is that the cohesin-CTCF are actually, so that CTCF, these domains arise because CTCF is translocating in the genome constantly. And so there's a pool of cohesin that is dynamic, and when it encounters a CTCF site, it will form what's called a CTCF anchored loop that shows up as a dot in these interaction matrices. So in the literature, frequently people call that loops. And the cohesin that is translocated and generating these interactions, it cannot cross the boundary because the CTCF sites are actually providing and blocking that cohesin from crossing that boundary. So the excitement in this field is because that might explain a lot of aspects of genome regulation, enhancer promoter interactions, insulation. There are links to RNA splicing, somatic recombination, for example, in immunoglobulins, VDJ recombination, also repair, imprinting, inactivation, etc. And so how does this machinery really catalyze that reaction was the key question that we want to set out. And the SMCs are these large ring-shaped complexes that are made up of called proteins, SMC proteins, that are bridged by a glycine, so they form a topologically closed loop. They are conserved, they even from bacteria to onwards, so they're even more ancient than chromatin itself. And the prevailing model is that DNA has to be captured within the loop of this cohesin molecule. Now, CTCF was originally identified at the milk locus as an insulator, and so it has the zinc finger DNA binding domains, which are used to recognize this CTCF binding site. But how CTCF and cohesin work together has been quite mysterious. Now, the cohesin or the SMC molecules, they form these ring-shaped complexes, and they're accessory factors that are important in catalyzing what's called loop extrusion. So the ADPase motor catalyzes DNA translocation, which is here shown as an increasingly long DNA loop. They're anchoring molecules that anchor the SMC protein to the DNA. And just recently we found out this is still a model for the condensin SMC, but we also found out that cohesin can also translocate on DNA and catalyze this DNA loop extrusion. Now at the head of the SMC molecules, if one goes back to this slide, they are ABC ADPase heads that need to come together to catalyze this reaction. It's a bit like the equivalent situation in ABC ADPases, where you have these nucleotide binding domains that need to sort of come together and sandwich ATP to catalyze, for example, transport of solutes across a membrane. So generally, carry out some biochemical reaction in in cohesin, this reaction is supposed to translocate DNA. The cohesin ring is shown again schematically here. It has a number of accessory factors. These are usually heat repeat proteins such as PDS5 and SCC3. And they frequently bind to the green SCC1, the bridging protein called glycine. It's called SCC1 in yeast. And here's the structure of this PDS5 SCC1 complex. And so there are a number of important regulators that are important in cohesin biology to regulate the loop extrusion activity, DNA engagement, etc. So the model is that when the SMC protein and the glycine molecule close and form a topologically closed structure by engaging the N-terminus of the SCC1, it can capture a DNA molecule. And this reaction is regulated by a loader complex called SCC24 that Stephen Hinshaw also worked on in Steve Harrison's group. And their release factors, for example, the protein called WAPL, releases the cohesin molecule from the DNA by presumably opening up this N-terminal N that attaches to the SMC proteins, and PDS5 and SCC3 contribute to that reaction. Now, we had a series of projects on cohesin, and here's one example. So we determined the structure of the SCC3 protein, which is shown in blue here, bound to the SCC1, and realized it's actually engaging DNA. And we determined a DNA liganded structure of this complex. And what we find is that this was a very challenging project because the DNA is actually sliding. These cohesin proteins, they're not DNA specifics and no specificity. And so the DNA substrate is actually sliding in the DNA binding pocket. And so it required a lot of crystallization trials and a lot of screening to find crystals that diffract to with reasonably good quality. And so one aspect of this work was that over the last couple of years, the automation on the beamlines has been pushed so much that even very challenging projects now, such as this one, where we had to really look at very many crystals, become more realistic. Now, what has been developed here at EMBL was a sort of a pipeline where you submit your protein sample, and then the rest is almost the entire process is automated. Once you have crystal hits, you click on your computer screen. You can do that remotely. It will get mounted into a loop. And then through a technology called Crystal Direct, it's cut out by a laser, mounted in the synchrotron, automatically focused through optical and X-ray centering, and then one can collect data in an automatic way. So, so that is the setup that we've used in this set, where one has the machine that harvests the crystals directly at the synchrotron beamline. It gets automatically, then once you identify the relevant crystals you want to look at, it gets loaded onto the sample changer, etc. Then it's optically centered, and in the second step, centered by X-ray centering. And so one can therefore, generate a much higher density of crystals, for example, and so look at many more specimens in an automated way. And so that allowed us to determine the structure of this SCC3, SCC1 subcomplex bound to the DNA, which is shown here in SCC3 in blue, SCC1 in green, and the DNA is engaging this, what we call a cradle, that is sort of, the DNA is sort of sitting in the cradle of this sc3 hook. And it turns out that this interaction is then critical for recruitment of cohesin to chromatin. Now, a key aspect of how this cohesin is regulated is that it engages the double, DNA, and it's important not only for catalyzing DNA loops, but also for holding sister chromatids together. But frequently, when people look at that by imaging, the complex is actually very dynamic. These coils are dynamic. And so what we did is we focused uniquely on this cohesin head initially. And we trapped, in a recent paper, we described how we trapped this cohesin head where much of the regulation is happening by generating a linked form of this complex, which allows us to co-purify this cohesin head module where the much of the regulation is happening. And this enabled us to study this complex by cryoEM. And here is now a structure of this cohesin head complex to quite high resolution for these sort of, the very small particle. And what we found is that the vast majority of these complexes, they are actually compatible with a structure where the N-SCC1 interaction was lost, and only a small fraction of these particles were compatible with the conformation where the N-SCC1 green part here is engaged. To remind you, opening and closing, that is binding and release of that green fragment here, is supposed to be critical for opening the cohesin ring complex. And so what we also did is, so this cryoEM structure was in the ATP sandwich state, so in the engaged or liganded state. But to compare that and to understand how ATP binding and complex formation, how the conformational changes associated with that reaction drive the remodeling of the complex, we also determined a crystal structure of the SMC1's subcomplex in the absence of the ligand of the ATP ligand, and so one could then compare the unligand and liganded form, and that's what we've done in this figure. So the gray one overlaid is the unliganded form, and then the liganded form is shown in red. And one can see that there are conformational changes that arise due to that heterodimerization and engagement of the ATP that are propagated through these 3 helices. And so there's an outward propagation of a movement that arises that leads to remodeling of these flanking coiled arms of the SMC complex. And so more specifically, there is concerted movement. A key glutamine rotates in and it engages the ADP magnesium complex, and that allows a helix to move in, this helix 3, and that's coupled again to this helix 4, which then propagates that movement outwards towards the coiled coil. So the movement that one then gets is that upon heterodimerization and ADP engagement, is that the coiled coil rotates outwards and hence clashes with a conformation that it otherwise has when it's not engaged. So that suggests directly that formation of this ABC ADPA sandwich and propagation of the conformational changes here leads to is coupled to opening of the cohesin ring by displacing by remodeling the epitope of binding of the SCC1 molecule. So we had a series of different structures on the cohesin complex, and then wanted to ask how does the complex engage CTCF, which is sort of a very mysterious process. And what a student did in the lab, Yan Li, she realized that that there's a fragment in CTCF that when she expressed proteins that have truncations of CTCF that have that fragment, she was able to, in pull-down style analysis, show that CTCF actually binds a component of the cohesin complex, and that's the SCC3 component that I already mentioned earlier. In mammals, it's called SA2. It's a yeast SCC3, or mammals SA2 or STAG2 also. And so this fragment of CTCF here, she was able to narrow it down to a roughly 10 amino acid peptide, was sufficient to engage a subcomplex. And so we call it SA2, SCC1 subcomplex with micromolar affinity. And so Jan was then able to crystallize this complex, and again, this automation of the beamlines, and was actually an important asset for looking at many of these crystals. And she was able to determine this structure of this ternary complex and found that CTCF, the fragment she identified, engages a composite surface on the SA2 molecule. That is, so where SCC1 and SA2 come together, CTCF engages into this composite pocket. Now, there are two key residues that are highlighted in red in this region. It's a tyrosine and phenylalanine. And it turns out that this interaction is actually critical for how TADs in the genome arise. Now, another interesting observation was then, when we looked at the mutations in cancer in this right quadrangle here are highly prevalent, frequently mutated genes in cancer by the Lawrence et al. Study, P300CBP, but also the STAG2 protein and CTCF are very frequently mutated in cancer. And when we plotted now this with mid-sense mutation frequency from Cosmic database onto our structure, there is a hotspot that is highly red, is this arginine 370, which maps right into that interface where CTCF is binding. And so that was quite interesting because it suggested that abolishment of this interaction by mutation is actually something that arises in disease. Now, Jan then confirmed that this interaction is, the usual mutations that one does to abolish this interaction in vitro, both on the CTCF side, on the SA2 side, or on the SCC1 side of this interface. Now, interestingly, what we also found from the literature was that there are other proteins, such as the release factor of cohesin, this release factor is called WAPO, also has similar motifs in its N-terminus. And so that suggested to us that CTCF might actually counteract this general cohesin release factor by engaging a surface on this SA2 protein. So in collaboration with Benjamin Rowland's lab, we generated specific mutations in just this key residues in CTCF. And then ask what happens to genome folding in cells. Turns out that these cells when you mutate. So normally CTCF is essential. You cannot delete it from cells. But the 2-point mutations were actually viable. Now, what we first observed is that if one compared the cells that have the point mutation and one looks at and compares them against CTCF wild type, we found that the mutated form, when CTCF cannot interact with this cohesin SA2 protein, has actually a much higher half-life when you in FRAP experiments. So when you bleach and then look at recovery, the wild type would recover more slowly, whereas in this, so we're looking at cohesin here. So cohesin is recovering more slowly in the wild type, whereas when you mutate CTCF, so CTCF somehow stabilizes cohesin on chromatin. And it turns out when we did biochemical competition experiments, we could show that that when you increase WAPL, a fragment of WAPL that has these FGF motifs, then you actually can disrupt that binding to CTCF so that it's directly antagonistic CTCF and WAPL binding to the SA2 protein. Now, when we then did this high C experiments, we realized that there are no, in left it would be a wild type high C interaction map, and in the right is the mutant form. Then what one can see is that there are no more CTCF anchored loops when you specifically mutate just those 2 residues. And when one then looks at that in the entire genome, one can find that actually all detectable loops are gone across the genome. That's shown in this, what's called an aggregate analysis, where one piles up all these different loops across the genome, and then one can see that in the CTCF mutant, all these loops are gone. The TAD structures, so if one does an aggregate analysis, there's still sort of residual TAD structure, but the corner peaks are largely disappeared. And so we then did additional experiments to show that the CTCF actually stabilizes cohesin on chromatin. And that's shown here. For example, it's a qPCR, chip qPCR experiment where one looks at the different positions at the CTCF sites that give rise to these peaks and these TADs in this part of the genome. And one can then generate primers and look at by chipping for SCC1, one can then show that in the mutant, you actually lose SCC1 as compared to the wild type, specifically at the CTCF sites, okay? And so overall, the chip sequencing shows that at the wild type sites where you have strong signal, in the mutant, it's usually decreased. So cohesin is not retained at the CTCF site in these mutant cells. And again, one can look at this in the entire genome. So the wild type is for cohesin is shown here, and there's a great reduction in the mutant cell line, whereas CTCF itself does not change in this analysis. Now, this so far suggests that that CTCF has a specific motif that is conserved, that interacts with the cohesin complex, the SA2 subunit, and directly counteracts the release factor WAPL. But it was reported there are additional FGF repeats in a set of cohesin regulators. And so what we thought is that there's probably an entire motif that we then generated, so what's called a regular expression motif to cover these different ligands, and then search the entire proteome to see if there are additional potential cohesin regulators. You could imagine that if you just look at these FGF motifs, they're actually everywhere. But if you just look at intrinsically disordered segments, which our structure shows is a requirement, and you look at conservation and you ask that it's nuclear, then you actually don't have so many potential ligands. And then we looked at that with peptide arrays, and here's just an example of that, where you probe that with against these these peptides derived from these potential new ligands, and we find that there's a set of ligands, they're indicated in green here, that we think might be bona fide SA2 ligands as well. And so these ligands are actually all interesting because, as I said earlier, you can see, for example, the CTCF would be a very strong binder here, which gives you a very strong dot, and then you mutate, in this case, just a hydroxyl group, and you weaken this site, this interaction, and additional mutations completely disrupt this. But there's a set of interactors that bind with almost equal affinity as the CTCF does. And some of these are important, for example, in replication. So the hypothesis is that maybe MCM3 is part of the replicative helicase. So is that interaction important to stabilize cohesin behind the replication fork to establish what's known as sister chromatic cohesion? Cohesion protection of the centromere might operate this way, as well as cohesion protection in meiosis. And so in summary, this reveals then the structural basis for how these loops are formed, because cohesin with the conserved essential surface here engages just the N-terminus of CTCF. And so when you model this interaction, there's only a small linker that is then linking that to the first zinc finger of CTCF. And so, in other words, cohesin can only approach CTCF from one side from the N-terminal end of CTCF, not from the C-terminal end. And we think that that is why in the genome, you have this convergence rule. You always need the two CTCF sides to point inwards, and that's where you form tails. We think that CTCF counteracts WAPL, and when you mutate that interaction interface, cohesin becomes more dynamic, and because WAPL constantly removes cohesin that is not stabilized by ligands such as CTCF. And so to recap, I told you about the head structure of cohesin, how that leads into insights into the gating mechanism. I talked about cohesin-CTCF, how that leads to insights into the genome folding reaction, but also how new ligands might use the same principle, how to regulate regulate cohesin's ability to catalyze folding by counteracting what we think is a constant release reaction. So in TADs, cohesin is constantly released, and these allow the intervening sequences not to interact with other sequences that have similar chromatin modifications. And so this is constantly happening, and there's a pool of cohesin that's dynamic. And that's why we get this dynamic loops that arise. They're stabilized at the edge of the TAD by CTCF through the interaction I just mentioned. To summarize, so just to thank people again, this is largely driven by Carl Muir and Jan Lee, my group. The collaborators already mentioned, and cryoEM facilities, HTX facilities, etc. And thanks for your attention.
Thank you, Daniel. So if we have any questions, I think maybe as I mentioned, to make it more interactive, people can just raise their hand or just unmute themselves and fire up, or potentially if you prefer to send me a chat message, you can do it that way as well. So one question, Steve, do you want to ask directly?
Yeah, can you hear me?
Very well, yes.
Oh, great. Yeah. Yeah, so thanks, a very nice talk. I'm wondering how your model for cohesin stabilization relates to the recent finding that SCC24 is actually required for the loop extrusion activity of cohesin, and specifically interested in how you might imagine the entire machinery comes together.
Yeah, I think that's an important point. So it looks like the SCC24 complex is important to stabilize a conformation that allows cohesin to translocate on chromatin. But the stabilized loops that arise at the CTCF side seem to require PDS5, which is mutually exclusive with binding SCC2. And so we do not know at this moment how this is switched over specifically at CTCF sites, but there's an additional interaction between CTCF and PDS5 that we know about. And so that is maybe one way.
Okay, thank you, Daniel. We have a question. Do you want to jump on and ask the question?
Hi, yeah, really nice talk. So I was actually wondering, in light of the model that you proposed, does this also somehow predict which CTCF sites would be chosen for, the looping interaction? Because there are definitely more CTCF sites in the genome that are in the convergent orientation, but not all of them are engaged in this looping.
Yes, I mean, you might know that the CTCF sites are frequently methylated, so it's also regulated by imprinting, and CTCF is also dynamic. So in the ensemble, when you look at millions of cells, you might have many sites that are actually not occupied, and that's maybe explaining that in part. But also methylation, of course, of the sites, and imprinting might explain that that observation. Does that make sense to you?